Thanks everyone for taking the time. It's wonderful to be here with so many people that are making a great contribution to science and clinical and regenerative medicine. We think of where we were just 5 years ago and think of where we'll be 5 years from now. It's pretty inspiring. So I want to tell you a little bit about Encoded Therapeutics, our pipeline and our platform. Encoded is established. We want to establish Encoded as a permanent leader in CNS diseases and genetic therapies, and they go well beyond monogenic disease. The company is well-funded with $350 million in cash that should get us to 2025, which is keeps us able to sleep better at night. We're a company of about 200 people. We have 2 locations, primarily in South San Francisco is where our research is done, and we just opened a manufacturing facility in North Carolina. Our leadership team comes from various companies with strong backgrounds in genetic medicines, including our regulatory officer, manufacturing officer, both came to us from Avexis, where they had some of the same roles. Sal Rico, who joined us from Audentis, where he was the head of development. And our chief technical officer, Martin Moorhead, who has a long history in everything genetics, along with our CEO, who was among the Kartik Ramamurthy, who was among the founders of Voyager, the founding team. So the way we judge our success over the next period of time is developing and moving ETX-101 for Dravet syndrome's our lead asset into the clinic in the first half of 2023. We aspire to have an IND in our CNS indications for every roughly 18 months. We want to continue to enhance our genomic and computational discovery platform, which was what Encoded was founded on in 2015. And we want to build best-in-class manufacturing. And of course, we want to do this through attracting and keeping some of the most talented people in the world working in genetic medicines. So I want to tell you a little bit about our lead asset is in Dravet syndrome. Dravet syndrome is a progressive epileptic encephalopathy with lifelong developmental impact and a high childhood mortality. These kids, it's a pretty sizable population. It's an incidence rate of 1 out of every 15,500. About 20% of these kids pass before they are, due to sudden seizures, before they are 18. The kids that continue or have a very high seizure burden throughout their childhood and never develop beyond childlike symptoms or childlike abilities from a neurodevelopment standpoint. So severe unmet need and opportunity for intervention. So our lead asset, ETX-101, is a AAV-based gene therapy that takes that transcription factor incorporates an AAV9 administered through ICV. We do this and we target directly into the brain in the SCN1A gene to upregulate the gene to produce increased NaV1.1 protein. We do this to get to normal physiological levels, so that's about a 20% increase, which achieves our ambition. What gives us confidence in ETX-101's success is its work in mice thus far, which if you look on the left, you see a mortality study that we did showing that ETX-101 treated mice had a long, durable life that was consistent with wild type, whereas untreated mice had a significant mortality rate that was well beyond 50% after 6 months. If you look on the right side of the, you see a graph that shows seizure burden, where ETX-101 treated mice had virtually no seizures until turned up to the most highest levels of temperatures, versus non-treated mice had very significant seizure burden. So this, along with other work, convinced us that we were onto the right track with SCN1A upregulation. The program itself has significant preclinical research. We moved last year into a natural history study in vision to further validate the burden of disease with these kids, and some of that work has actually shown that despite newer treatments for seizures, these kids carry still a significant seizure burden, and as young as 6 months to a year, they're starting to fall off their neurodevelopment trajectory compared to their neurotypical peers. So this program, we are in the IND stage now. We've submitted our IND, and we expect to be into the clinic first half of next year. Moving more toward the pipeline, we have 2 programs that are in monogenic disease that are not disclosed yet, but we expect to move those further into tox studies and do candidate selection early next year and move further into tox studies. We're also looking at non-monogenic populations in refractory epilepsies, so where we can use a genetic target to improve seizure burden on these on non-monogenic populations. That's pretty interesting work, and it's really an opportunity to get into much larger populations where nothing has really been able to be effective. Can't talk about AAV gene therapies without talking about manufacturing. I agree with my predecessor that process development is a critical success factor in the very beginning. Encoded's invested in process development pretty early on its time. Our current processes, manufacturing creates a yield that we believe is best in class from an AAV standpoint, both in yield and regulatory. We have the manufacturing facility that is currently doing non-GMP production and should do GMP production by the end of next year. This will make us independent from a clinical standpoint, non-clinical and clinical, and then over time we expect to go to commercial scale. So shifting away back to what really made Encoded different and what it was founded on, Encoded at its heart is a gene therapy engineering company. So we look at our platform as of enhancers, different regulatory elements, including enhancers, promoters, and look at what we can affect transgenes. And what we focus on is doing, whether it's cell selectivity, whether it's titratability, whether we're looking at gene replacement, upregulation, or silencing, all different aspects of the gene therapy engineering standpoint, and using clinically validated capsids to achieve our goals. And some of our target product profiles, for examples, include where we're looking for cell selectivity, which is what we're using in ETX-101, which targets gabatory inhibitory neurons, and whether it's potency, which you see in the middle, where we had a liver program that had very small regulatory elements to replace a full large transgene and increase significant levels of potency, are some of the work that we've done recently and is currently being shown in ESGCT this week, is detargeting, where we are looking at achieving our expression profiles in CNS while also minimizing and getting virtually very little expression in both liver and DRG, which we believe that those two detargeting aspects together open up a lot broader therapeutic window for a lot of different opportunities. Finally, we are looking to use our platform not only to envision our own pipeline and lead asset, but looking for partnering in areas outside of pediatric CNS. So with that, that's it. If there are any questions, I'd be happy to take them. Thank you.